So in the upcoming lab, we're going to be replicating analyses from the Curran and Doyle paper, looking at EEG as it relates to different kinds of memory effects. And so we're going to be using the CD2010 script that we ran on the first lab day to replicate those analyses. So instead of running it all at once like we did last time, this time we're going to be running it piece by piece. So the first thing I'll show you is that if you click on these different sections, it will highlight certain parts in yellow, and then you can run it piece by piece instead of all at once. And the way that you can run a section at a time is once you have highlighted a section in yellow, then you would click Run Section. And you know it's done when it stops saying busy in the corner. So the other thing I want to show you in this tutorial is just the different kinds of figures and graphs that are going to be popping up as you run this script. As an example, when I run this part Q1, I'm going to get a figure like this. So what's happening here is each of these squiggly lines is the recording from a different electrode over time. And so we can see this a little bit better if we zoom in, and instead of looking at all of the electrode channels, let's just look at 10. And so now you can see, for example, here, E118 is the electrode 118, and you can see recording over time. Down here it's going up to 5 seconds, but we can scroll through using these arrows. And so this is basically all the raw data for one subject from all of the different electrode channels. Okay. Another kind of figure that's going to pop up is something like this. So this is showing something called ERPs, which stands for event-related potentials. And so what that means is it's looking at the average signal following a stimulus. So that means that for that participant, we're looking to see what does their activity normally look like for a couple seconds after they're shown a new stimulus, whether it's a picture or word. And it's showing that signal, that average signal, at each of the different electrode locations. So this data is for one individual subject, and we're looking at the average signal following stimulus at different locations. And you'll see that this data becomes a lot nicer looking once we average across subjects as well. And here we can see these more distinct patterns. And we can also click on one of these. For example, I'm going to click on E116, and that'll bring it up in a bigger window. And here we can see the black line represents the word condition, blue is for picture, and red is for the new. And the last kind of figure that I want to preview here are these topographic maps. So this is showing the change in electric signal mapped over the brain. And so the different numbers represent the different electrodes. And if you click on these brains, you can actually rotate them. So here we can see the front of the brain, and the red areas are going to indicate a more positive change in voltage, whereas these darker blue areas indicate a more negative change in voltage.